Okay, so Streptococcus. We are done with Staphylococcus. Now we go to Streptococcus. I can use this table. Hope you can see here. So we know it is gram-positive. So remember, when you get a bacteria, you've found it's gram-positive. You identify, is it a bacillus? Is it a coccus? If it is coccus, it can either be two. What do? The catalase test. If it is positive, it is Staphylococcus. We are done with that. If it is negative, it is Streptococcus. And usually it occurs in chains. Can you see here? It can either be in chains or in pairs. These ones are always in clusters. And then the next step is we grow it in blood agar. When you grow it in blood agar, there are usually three outcomes. It can either be beta-hemolytic, alpha-hemolytic, or gamma-hemolytic. If it's beta-hemolytic means there is complete hemolysis. Okay, and it's usually clear. Okay, and that happens, you can have two types of bacteria. We have pyogenes or group A strep and Streptococcus agalactiae or group B strep. Then how do you differentiate between these two? We use something, we call it bacitracin test. Okay, if it's bacitracin sensitive, it is group A, strep pyogenes. If it's bacitracin resistance, we say group B or agalactiae. Okay, if it is alpha-hemolytic, this means that there is partial hemolysis. It's usually color green. We do so there are two types of bacteria that will be alpha-hemolytic, pneumoniae and virulence. After that, we do the optokin test. For optokin test, what happens is optokin sensitive, strep pneumoniae. Optokin resistance, strep virulence. And then for gamma, basically there is no hemolysis that is going to happen. We usually have Streptococcus. There are quite a lot. Fecalis, bovis, enterococcus. Okay, we're going to look at that. So what I want you to remember, these two can be really confusing. For this, the formula is usually BB-RAS. Okay, BB-RAS. So the B is, you say bacitracin, group B resistance, group A sensitive. BB-RAS. Okay, bacitracin, group B, BB-RAS resistant, group B resistant, and then group A sensitive. That's how I usually remember. BB-RAS, that's usually the formula. And then for alpha hemolysis, I usually say overpass. Over. So it's OVRPS, overpass. So overpass, that is optokin, the optokin test, virulence resistant, and then pneumoniae sensitive. Like that. BB-RAS and overpass. That's how I usually remember, because these two can be really, really confusing. And this is very, very high yield, okay? Because they can bring this picture, and then they tell you what is the next test to be done. They can bring you this picture, they say, what is the next test to be done? What do you expect if the lab tech cultured group B Streptococcus then you have to say bacitracin was done and it was resistant, like that. Okay? I hope we've understood. Okay. Any question on that? BB, RAS, and overpass? So Streptococci is gram-positive spherical stroke ovoid cocci. So cocci means oval. Arranged in long chains, commonly in pairs. So they'll be in pairs and then they are in chains. Classification of Streptococci includes classification system is based on hemolysis, beta-hemolytic, alpha-hemolytic, gamma-hemolytic, as we had seen in that chart. And then we have something called as the Lancefield classification of beta-hemolytic Streptococci. So we have group A, B, C, D, 17 groups. The most relevant is group A and B. Okay, so it is classified based on the carbohydrate antigen in the cell wall. So as you can see here, beta hemolysis is clear, alpha is green, gamma, no hemolysis will take place. Okay, so these are the most important. We're going to learn about them. We start with the general characteristics of all the streptococci. Number one, it is gram-positive. It is a cocci arranged in long chains, commonly in pairs. Number two, non-spore forming. So the only two, what are the only two spore forming bacteria that I told you last time? Answers in the chat. The only two spore forming. And what is the function of a spore? What is the function of a spore and what are the two non-spore forming? Sorry, the two spore forming bacteria. And then it's also non-motile. Can form capsules. Okay. Facultative anaerobes meaning that they can use oxygen or they require oxygen or they may not require oxygen. Most parasitic forms are fastidious and require enriched media. Okay. Answers in the chat for the spore forming. So when we say fastidious, it means that we're going to require complex nutritional requirements. So as you're going to see. And then small non-pigmented colonies sensitive to drying, heat, and disinfectants. So we start with the strep pyogenes. There is no answer. The two spore-forming bacteria I mentioned when I was talking about spores. Clostridium and E. coli. No. E. coli do not form spores. So it's Clostridium and Bacillus. Okay. And they are both gram-positive. Then the function of the spores, basically to survive harsh conditions, be it heat, chemicals, any environmental stressors. Okay. Strep pyogenes, most serious streptococcal pathogen. It's a parasite and it inhabits the throat, nasopharynx, occasionally the skin. Gram-positive, spherical, non-motile, can form capsules. So this is strep pyogenes, also known as group A strep, which is very, very important. This is also a very high yield, okay, type of bacteria. And what I want you to remember is, what I want you to remember from this is it affects, so it inhabits the throat, nasopharynx, and occasionally the skin. So the pathologies that will occur in these areas where the bacteria is found. So facultative anaerobes, best grown pH of 7.4 to 7.6 and a temperature of 37, required enriched media for growth. The only bacteria that I have seen mentioned where you have to know the temperature is a bacteria known as Campylobacter jejuni, where the temperature for growth is 42 degrees Celsius. The rest, no one cares. Okay? Note that down. Campylobacter jejuni grows at 42 degrees Celsius. That's the only one I've seen asking about temperature. The rest, not a must, you know. So as you can see here, the colonies are around 1 millimeters in diameter, surrounded by a zone of clear hemolysis, semi-transparent and vary in appearance. Can you see here? So I told you when you see hemolysis, usually clear. Look. So lysis has occurred, hemolysis has occurred here. These are strep pyogenes. Then biochemical properties produce many virulence factors, i.e. production of enzymes and toxins. So let's look at them. Number 1, streptokinase. So don't confuse this with staphylokinase. Digest fibrin clots. Hyaluronidase breaks down connective tissue. DNase hydrolyzes DNase. You see, these two are similar to Staphylococcus aureus. And then the toxins produced streptolysin or hemolysin, streptolysin O, SLO, and streptolysin S, both cause cell and tissue injury. This is very, very important because there is a test we do. We call it anti-streptolysin O. Antibodies against this, we look for them when we are testing a certain disease caused by strep pyogenes. Okay? So this is very, very important. Erythrogenic toxin or pyrogenic. Anywhere you see pyro, this means fever. So fever is also known as pyrexia. So pyrogenic meaning causing fever. Okay? We usually call it scarlet fever. Then the super antigen, strong monocyte and lymphocyte stimulant cause the release of tissue necrotic factor. Those are the toxins produced. Then pathogenesis, humans are the only reservoir and apparent carriers, visible carriers of toxins and enzymes. Transmission is through contact, droplets, food, and fomites. So as you can see, the throat, nasopharynx, and the skin. So how will it be transmitted? If it's found in the throat, when you cough or when you sneeze, you can get it. Contact through the skin. Food, maybe food poisoning. Okay? Then fomites. So fomites are things like, you know, utensils, bedding, clothing. Okay? Portal of entry is generated through the skin or the pharynx. Children are predominant group affected for cutaneous and throat infections. Systemic infections and progressive diseases may occur if untreated. Now, these are the diseases that it causes. But I want to classify them so that you can understand. We can classify them in terms of what toxin caused it. So generally, there's impetigo, throat infections, rheumatic fever, and acute glomerulonephritis. But in order for you to remember, we can classify them into 3. Number 1, pyogenic. Okay, so pyogenic, we'll put throat infections, impetigo. So when there's impetigo, you can also have something called cellulitis. Cellulitis is also an inflammation of the skin. Okay. And then we can classify it in terms of toxigenic. Toxigenic is now caused by these toxins. Okay. So the toxins usually cause so remember, in Staphylococcus, we caused something called toxic shock syndrome. Okay? But in Streptococcus, because it is not exactly the toxic shock syndrome, we call it toxic shock-like syndrome. Okay? So it causes something called toxic shock-like syndrome. Quite similar to toxic shock syndrome, but now the causative agent is strep pyogenes. And then the third classification is immunologic. Immunologic now, it's rheumatic fever and acute glomerulonephritis. Okay. This is the impetigo that I was saying, as you can see on the skin. And then these are the rashes. Normally, it causes something known as scarlet fever. So scarlet fever is where you have a sore throat. Then after the sore throat, you have these rashes. Okay, so it's usually a patient who has a sore throat, then they get rashes, usually known as scarlet fever. And then as you can see here, the rash on the face in this child, and then pharyngitis, which is inflammation here. Okay, then for these, because I don't think I've explained them here, that is impetigo, you've understood. For the immunologic, so what happens is in this case, why rheumatic fever will occur is you start with pharyngitis. You develop pharyngitis or a sore throat. So we call it a strep throat. Then what happens is you see when your body is fighting this bacteria, a certain protein is known as the M protein, okay? So the M protein is one of the virulence factors in Streptococcus pyogenes. Now this M protein, it goes and it is similar, okay? So you learn in immunology something known as molecular mimicry. This M protein of this strep pyogenes is similar to the protein found in our heart valves, okay? Now the immune system, since it's attacking this M protein with its antibodies, it is going to get confused because it will be like, there are M proteins on the pharynx, and there are also similar things that look like M proteins on the heart valves. So it's going to attack the heart valves, okay? So that is what we call molecular mimicry. It is being confused. Now what happens is, for a child with rheumatic fever, it's a very dangerous disease. They get a throat infection, then maybe 2 weeks later, they come with a heart issue. Now that you'll know that that is rheumatic fever. Okay. Have you understood? Now, similar to the acute glomerulonephritis. So glomerulo, this comes from the glomerulus in the nephrons. Then itis means inflammation. So acute glomerulonephritis, normally it's usually called post-infectious, post-streptococcal glomerulonephritis. This is because now in this case, the antibodies that were targeting your skin, remember you got impetigo, then there are antibodies on the surface of the skin attacking those cells in the skin. But then 2 weeks later, you get a kidney infection. This is because the toxin produced the toxin produced, that same M protein that was produced in your skin is similar to those in your kidneys. So the immune system gets confused and it's like, oh, so I'm supposed to attack this strep pyogenes M protein on the skin, but it's also similar to the one in the kidney. So now you get a kidney infection. Are you understanding? Or should I repeat? Are you understanding?
Repeat.
So the pattern of illness is this. A child will come in with a skin infection. You treat it. After 2 weeks, they come with a kidney infection. That is the first case scenario. The second case scenario is a child will come with a sore throat. You treat them. 2 weeks later, they come in with a heart issue. So you're asking yourself what is happening. Strep pyogenes is producing something known as M protein. And the M protein, it's a toxin, okay, that is produced by strep pyogenes. And the function of this M protein is just to inhibit phagocytosis, okay? So what happens is the body is trying to destroy this M protein, but this M protein is structurally similar to the heart valves and to the glomerulus in the kidney. Okay, so these M proteins, they are different. Okay, so different species have different M proteins. So like the strep pyogenes, it produces different types of M proteins. So it might produce M proteins that are similar to the throat ones, that you'll get a throat infection, and similar to the heart, and when you get a throat infection, and then the M protein produced when you get a skin infection is similar to the one of the kidney. So when your body is clearing this M protein of the skin, it is similar to the structures in your glomerulus, which are found in your nephron. So what happens is there'll be an immune response that is going to go to your kidney because the body is confused. It is similar, it is mimicking. Okay, that is why we call it molecular mimicry. Okay, then for the strep throat, the M protein produced when you got the throat infection is similar to the structure in the heart valves. That is why you have extensive heart valve damage. Okay, that is what we call rheumatic fever. But this is something you'll expound more in immunology. I just wanted to mention as you do immunology too, but this is something you should know, okay? And it is caused by strep pyogenes. Okay, that is more of pathology. In microbiology, they just expect you to know impetigo, rheumatic fever, strep throat, and acute glomerulonephritis. But in pathology is when they want you to know the details. Okay. I hope I'm clear. Then here are the pictures. Then for the lab diagnosis, we have the skin. So you're going to take a skin sample, a throat swab, sputum, urine, and blood, because this is the sections where the strep pyogenes is infecting. Then the different methods of lab diagnosis. So gram stain, we have culture and catalase test. So as you can see here, gram stain, we know it is gram-positive, cocci in chains, as we had said. And then the culture, it will be beta-hemolytic. Okay, so it will appear clear. Then bacitracin, so we said BRAS, bacitracin group B resistant. So it will be resistant. And then we have the catalase test. Normally how it's done is you transfer a small amount of bacterial colony to the surface of a clean, dry glass slide using a loop or sterile wooden stick. Place a drop of 3% hydrogen peroxide onto the slide. So you place it there. A positive result is the rapid evolution of oxygen with 5 to 10 seconds as evidenced by bubbling. Okay, so normally the enzyme catalase, it breaks down hydrogen peroxide to water and oxygen. So you're going to see these bubbles. But we know that strep pyogenes is catalase negative. The only catalase positive is on the other side of the table, which is the staphylococcus. Here, you see catalase test negative, but here for staphylococcus will be positive. So yes, and then treatment. So strep pyogenes is one of the few bacteria that is still treated with penicillin. And then if you're allergic to penicillin, you can give erythromycin. Okay. I hope we're clear on strep pyogenes. So prevention, long-term penicillin prophylaxis for people with a history of rheumatic fever or recurrent strep throat. Then strep pyogenes, infection are best prevented through effective hand hygiene. I hope we're clear with Streptococcus pyogenes. Okay. Streptococcus pneumoniae. I should have started with, didn't do it. It's okay. Maybe just to mention on, before I go to Streptococcus pneumoniae, on Streptococcus, this group B, Streptococcus, it's called Streptococcus agalactiae. So in agalactiae, what happens is group B. What I want you to remember for Galactiae, B for babies. Okay, group B for babies. So why is it for babies? Is this because it mainly affects babies? It causes pneumonia in babies, meningitis, and sepsis. Pneumonia, meningitis, and sepsis in babies. So what happens is, it's a very high-yield thing you should know, is normally when parents, mothers are 35 to 37 weeks pregnant, they have to be screened for this group B Streptococcus. So usually take a vaginal swab, send it to the lab. In case they have it, you start giving them penicillin. Okay, because we don't want the group B Streptococcus to be spread to the baby. As we've said, it can cause pneumonia, meningitis, and even sepsis. Okay, that's just those two facts. If you know group B is for babies and mothers are screened before they give birth and you give penicillin, that's enough. Okay. And then we go to Streptococcus pneumoniae. These are commonly seen in the nasopharynx of healthy persons. From the word pneumoniae, it will not cause any illness itself unless a viral infection or other factors provokes it. So as you can see, these are the general characteristics of the bacteria we had said. But they're small, oval-shaped, arranged in pairs and short chains, as you can see here. Then cultural, it can grow in blood agar or chocolate agar. Growth improves by 5 to 10% carbon dioxide. Facultative anaerobes, best growth achieved in temperatures of 25 to 40. Colonies are surrounded by greenish hemolysis under aerobic conditions and clear under anaerobic conditions. So on our table, let's go back to our table here. Pneumoniae is alpha-hemolytic, so it will appear green. And then optokin sensitive. So from this sentence, they're saying colonies are surrounded by greenish hemolysis under aerobic conditions and clear under anaerobic conditions. So in room temperature, there's oxygen, so they'll appear green. But in anaerobic condition, they'll be clear. And then they don't really produce toxins for strep pneumoniae. In fact, the most important thing you should know is they cause pneumonia. I think I had written, yeah. Presence of some of the extracellular products, hemolysin, immunoglobulin A1, protease, neuraminidase, hyaluronidase. They like catalase and peroxidases, ferment glucose, lactose, and sucrose with production of acid. Have you seen this point? I want you to remember. IgA protease, immunoglobulin A1 protease, okay? They produce that. And they are capsulated. Remember that. Strep pneumoniae, capsulated, and IgA protease. And then pathogenesis, 5 to 50% of all people carry it as normal flora in the nasopharynx. Young children, elderly, immunocompromised, those with other lung disease or viral infections are at risk. Pneumococci multiply and induce an inflammatory response, gains access to the middle ear by the way of the eustachian tube, especially in children. It causes something known as otitis media, or infection of the middle ear. So it can cause pneumonia, infection of the lung parenchyma. So normally it causes lobar pneumonia. When we say lobar pneumonia, we mean that it causes pneumonia in a specific lobe, and then otitis media, especially in children. It can also cause meningitis and sinusitis. Sinusitis is inflammation of the sinuses. Meningitis is inflammation of the meninges. Okay. Then before I mention this, okay, I think I'll mention that. So lab diagnosis, strep pneumoniae is frequently isolated from samples such as sputum, blood, wound, and CSF. Remember I said it can cause meningitis. So cerebrospinal fluid is important. Then different methods of lab diagnosis: gram stain, Quellung test, culture, and catalase. What did we say the Quellung test is for? I had mentioned last time. What is the Quellung test for? Someone type in the chat, but it's actually here. Let me skip, you'll tell me. Culture. The organism is cultured on blood agar or chocolate agar with incubation and environment of carbon dioxide. The organism is cultured on blood agar with an added octokin disc to show alpha-hemolytic colonies. So it is sensitive. As you can see here, the octokin has killed the bacterial colonies, so meaning it is sensitive. If the colonies still grew here, we'd know it's resistant. Then catalase test, we know it's catalase negative. What is the Quellung test for? Let me give you a hint. Look at this. What is this white? The whitish part. What's the Quellung? And I remember I had mentioned, what did I say? So we treat with penicillin, G or V, broad spectrum antibiotics, so the erythromycin is what we give for patients who are allergic to penicillin. And then we have vaccines available: capsular antigen vaccine and the conjugate vaccine. So the reason Quellung tests whether the bacteria is encapsulated. Very good. As you can see here, on this, you can tell that there is a capsule surrounding this bacteria. This is Quellung positive. But it was a test, it's no longer done. Okay, but they will ask you, the following are Quellung positive bacteria except, okay, so you have to know strep pneumonia is Quellung positive. So one thing I want to mention here is the reason we give vaccines is because strep pneumoniae is cleared from the body via the spleen. Okay, so what happens is the macrophages go engulf the bacteria, then take it to the spleen, then it is cleared through the spleen. Okay, now there are some patients, let's say for example, if you've been done for a splenectomy. A splenectomy means that your spleen has been removed for one reason or the other. You won't be able to clear this bacteria, so we have to give you these vaccines. Another reason is patients who have sickle cell disease. Okay, I don't know if you've heard of sickle cell disease. For sickle cell disease, what happens is they usually have a spleen, but their spleen has become, in quotes, useless. We call it functional asplenia. Okay, it is no longer working. So in these patients, if they're infected by strep pneumoniae, they can get serious infections. So that's why we have to give them these vaccines. Okay, are we clear? So in high-risk individuals, these are the sicklers and the people who have been done for splenectomy. Then we go to virulence from the mnemonic or the mnemonic overpass. So octokin overpass, virulence resistant. So alpha-hemolytic, oval in shape, found in short chains. There are 6 species of virulence. The most common that you'll be asked in the exam is mutans. The rest, not relevant. Why I say that is because mutants, this one, mutants is the most common cause of subacute endocarditis. Why is it called subacute? Remember, for staph, it's called acute endocarditis. Acute because it occurs within a short period of time and the symptoms are very severe. Why this one is called subacute is because it causes less of a disease in the patient. Okay, so it will cause endocarditis. A good thing they'll tell you during exams is the patient had a dental procedure, because these things occur as normal flora in the gums and the teeth and the oral cavity. What will happen is you'll have a dental procedure. The Streptococcus mutans will enter the blood, find its way to the heart. And if you had a defective heart, let's say you had a heart valve, okay, or you had a defective heart, let's say you had rheumatic fever, or you had, let's say you have something that is wrong with your valves, the bacteria will go, it will form vegetations on your heart, then you're going to get endocarditis. Okay, have you understood? So we start with a dental procedure. The mutants, because it's a normal flora on the mouth, enter the blood, goes to the heart, causes endocarditis. So persons with pre-existing heart conditions should receive prophylactic antibiotics before surgery or dental procedures because of this strep virulence. It causes dental caries, sepsis, and endocarditis, but it's usually subacute, meaning that it's not a huge infection that will come with bad symptoms. Okay, so less pathogenic, if I was to use that word. Okay. Yes, I'm done with strep virulence. And then strep ficalis, also known as enterococci. So for enterococci, what I want you to remember is, yes, it's gram-positive oval cocci in pairs or short chains, identified by McConkey agar. What did I say about McConkey? What is the use of McConkey? Someone type in the chat. What is the use of McConkey? Colonies are magneta in color. I mean, magneta is, it's just a fancy way of saying pink. Okay, pink, pinkish, purplish in color and pinpoint. What is the function of McConkey agar? I had mentioned, someone type on the chat. It can grow in the range of 10 to 45 degrees and survive at temperatures of 60 degrees for 30 minutes. It is non-motile, facultative, anaerobe, ferments glucose, but does not produce catalase. It is associated with UTIs. So, okay. Remember I said saprophyticus and enterococci. These are the nitrite negative UTI bacteria that cause UTIs. Biliary tract infection, septicemia, endocarditis, and intra-abdominal abscesses. Aminoglycosides are the drug of choice. Normally, it causes nosocomial UTIs. Nosocomial means that it is hospital acquired. Yes, before we close, what is the function of McConkey? I've mentioned it. I mentioned it there. Lactose fermenters, yes. It is used to differentiate between lactose fermenters and non-lactose fermenters. And I had mentioned, I said that it is normally used in gram-negative bacteria. But now, in this case, enterococci is a lactose fermenter, so it will turn pink in McConkey agar. Okay, and with that, I am done. Any questions?